Welcome to Uncaged, the show that celebrates thought leadership from today's top business leaders. The program provides a voice to amazing executives from around the globe who are shaping the world of business today and mapping the path to the commerce of tomorrow. Today, we're speaking with Mario Bourdon. Mario, how are you?
I'm great. Great to be talking with you, Brent.
Yeah, and I'm very excited to talk to a fellow Dookie, which is also great, but also the Chief Scientific Officer of BioLaurus, which is innovating the future of in vivo preclinical molecular imaging by combining study design consulting, study management, and image analytics. And we'll go through the BioLaurus story that Mario has taken from the ground floor all the way to where it is today. But before we go there, Mario, let's talk a little bit about your background and your career.
Well, my background is in biology. That's an interest that's started out very early. Among my first memories, about 4 years old, crawling through the backyard on all fours, following a trail of ants. And that was, that kind of backyard safari. Where were they going? Did they come from? What was going on?
Right.
And that's been a thread throughout my life is learning new things, getting excited about discovery. And all of that has been focused through this perspective of how do we connect things and put it together in space and time. So from that early interest in how things happened, I had grew up here in Sacramento, California.
Lucky you.
When I was about 13 years old, my family moved down to San Diego. I went to high school here, and then I went to UCSD as an undergraduate. I was working my way through school, and I had my own little business, gardening, as a service here in La Jolla and Point Loma. And then I had this really great break. I got a job as a technician at the Scripps Institute of Research Institute, and in a laboratory of Frank Dixon, who was the director of the institute at that time.
Mm-hmm.
And it was an incredible experience. They were doing cutting edge research in autoimmune disease. They were doing new technologies, and I was so excited about it. They had a variety of techniques, including what are called radioimmunoassays. They were subsequently developed, ELISAs took their place as technology tools for measuring small amounts of drugs and biomarkers for various diseases.
Mm-hmm.
And from that experience, and again, the opportunity to do more, one of the things that came out as a characteristic of mine was whatever I was doing, I asked, couldn't we do it better? Couldn't we do it faster? Could we do it more efficiently? What would it be like? We had this regular structure of doing the assays, and we'd add one chemical part of the component of the assay, and we have to wait a little while for it to incubate. Right. And then we would go to the next step and the next step. And it was very much organized, had a very precise way in which we needed to do it.
Mm-hmm.
And I thought, well, couldn't we do a bit more? Well, one thing was going on, we could start another one, and they would all go in parallel downstream. And
yeah.
At the end of the day, we'd do more. I thought this was a great idea. My others in the lab didn't quite see it that way. It was like, Mario, why do you want to do twice as much?
Yeah.
We're getting paid the same. No, that wasn't the point. So, from there, I had a great opportunity to then grow and develop. Went to Duke University, and that was the thrill of a lifetime. I was there at a time when biomedical research was taking one of these great leaps forward. And I was really fortunate to be picked up as a PhD student in the laboratory of Daryl Bigner, who was an immunologist and neuro-oncologist. He's just an incredible scientist.
Yeah.
And so I was one of a couple of graduate students in that laboratory. And it was at a time where new technology, monoclonal antibodies, was just coming on the scene. It had been developed, first published about maybe 3 or 4 years earlier. And my task as a graduate student was to learn this new technology and develop monoclonal antibodies to human glioblastoma tumors. These are brain tumors.
Yeah.
And I had great success with that. We developed a monoclonal antibody that specifically targeted the glioblastoma tumors. And we looked at the biology of that. Where was it in the tumors? What kind of tumors? And all with an eye of the Bigner lab looking toward development of therapeutic. And that was Neuradiab. So this was a monoclonal antibody with a radioactive molecule, iodine molecule, that was intended to go into patients. In fact, it went all the way into phase 3 trials.
Wow.
To be able to treat as a therapeutic, immunotherapeutic brain tumors. So that was very exciting to be a part of that. Then did my postdoctoral work in what was then called the La Jolla Institute for, it was the La Jolla Cancer Research Foundation, Previs Medical Institute now. So there continued to learn about molecular biology, how to clone genes. And we cloned, among the first was I cloned a proteoglycan core protein. So you've heard of molecules like chondroitin sulfate. They're really important in how our tissues function and they change during development of tumors. And went on from there to a career in research, first at La Jolla Cancer Research Foundation, and then went into several organizations that were research institutes that had been created by for-profit biotech and pharmaceutical companies.
Right.
So this was at a period of time when the pharmaceutical companies and technology companies wanted to be able to support and grow basic research with the thought that they would have then the ability to access and target their own research and drug development more effectively. So that was an enormous opportunity to learn this collaborative environment and how we would do basic research and then, pass it on, pass the baton to those who would develop products and services and drugs to treat human disease. So I went to from California Institute for Biological Research, which was a Strategene company institute set up by Strategene, to an institute first set up by a unit of Pharmacia, which is now Upjohn. And And that was the La Jolla Institute for Experimental Medicine. And I was recruited to head up that research institute. And so that was very exciting. I was, still a young scientist, and this was a huge challenge, and risk. And I took it, and it was an exciting 15 years there.
That's amazing.
At somewhere along the way, Mario, you perhaps got the entrepreneurial bug. Maybe there was an opportunity, but you founded BioLaurus. So that happened, I think, in 2009. Tell me a little bit about how that story shaped and where you guys are today.
Well, it was serendipity, really. I mean, I had been involved in various aspects of molecular imaging all the way back to my graduate days at Duke, where we did monoclonal antibody imaging, and this was part of the unique situation of doing this fundamental research in a medical research department. So with that in mind, I was looking to set up a biotech company to develop some of our research discoveries as treatments for cancer.
Mm-hmm.
And as I was doing that and framing the organization, how would this company be formed? What resources would we need? How would I put together the program to be able to go out to the VCs, venture capital community for funding?
Mm-hmm.
And I came across a couple of companies that were doing something that I thought was amazing. And there was GE, for example, who was producing new instrumentation that had just come out in the past year for molecular imaging. And so these were SPECT instrumentation, there was ultrasound, there was CT. There was a variety of different technologies for this molecular image. It was like tricorders, right? The whole idea, just like you would do in the clinic, is you'd be able to look at where molecules were going in the body and where the specific things were happening and where inadvertent and unintended things were happening in the body. But you knew it in real time, which was far more, and in vivo. Really, really exciting. Here were tools that were going to make it possible to do things that we had never done before. It was like, Star Trek medicine now coming into our hands.
Yeah, like ideas that you had heard about and said, "Man, I wish we could," and suddenly, boom, it's right there.
Yes. And so here was transformative, disruptive technologies. Here's, we often take drugs and we translate it from the world of the test tube, petri dishes, and mice to man, into the into the clinical research. Here was an opportunity where it went in reverse, because all this technology was there and for human research, right, and been used for years. But now the instrumentation was there to be able to do this discovery research. So I was just excited by how this was going to happen and the potential for its utility. And so we've been able to work with a large number of primarily big pharma and large biotech companies on a whole array of projects. And this was something that excited me too. I was in, had my career developed in oncology, but here was an opportunity to learn about and work with exciting scientists from a whole range of diseases that had nothing to do with oncology, all connected by the interest in being able to see where drugs were going, what were they doing, the pharmacokinetics of these diseases. And we got into, in a big way, CNS diseases. So
that's incredible. What started as perhaps an insight and something that you saw an opportunity in 2009 still flourishes and grows today. And certainly I can see the role that you're able to play, in particular right now, in that obviously in vivo preclinical molecular imaging space. But I mean, tell me, Mario, more broadly, you've been a part of really the evolution of a lot of the research, the biomedical research, obviously biotechnology. You've been a part of helping redefine the drug discovery process and the development process. Where are we right now in all this?
Yeah, that's a good question. The developments of biotechnology is always seems to be in fits and starts. We go through long periods of doing much the same things.
Mm-hmm.
And then it takes time for various new tools and approaches to allow us to ask new questions and to get more answers that are more and more relevant to what is going on in our human bodies. Right now, I think there are several things that are going on, and particularly in one disease areas of central nervous system, the brain. Mm-hmm. The knowledge and the detail of what is going on, the incredible complexity of the human brain, and how that affects how we may approach new therapeutics is evolving really rapidly. COVID had a huge negative impact on research. We had our own research. We couldn't get into our laboratories, right? We were shut down for nearly a year. And
I'm excited to talk to you about that topic, because I think that from the general public perspective, COVID was a fascinating medical moment, certainly horrendous in terms of how it devastated people's lives. But at the same time, from a medical breakthrough perspective, wow. I mean, what an incredible story. We went from sitting around twiddling our thumbs, not knowing what the hell is going to happen, to suddenly rolling out vaccines around the world at light speed. I mean, I hope that we look back at this moment as kind of I guess maybe a research moment, a biotechnology rollout moment, a vaccine rollout moment that we can learn from. But it did also strike me that perhaps a lot of other areas of research might have been perhaps lowered in their priority in that moment, and perhaps slowed down. And I'd just be curious to hear, your experience over the last couple of years.
Well, as a provider of contract research, services, and particularly because we're on the really high end, high technology niche, that COVID has had a profound effect, and we're coming back from it, but we're coming back from it slowly. So yes, there was a huge impact across the board for most research, and it's taking a while for that to reemerge. So any disruptions, any problems in the world as a whole, right? It has effects on funding. It has effects on prioritization of research. And so it's quite a mixed bag. There are areas that are evolving with light speed. And again, for example, the mRNA approach to vaccine development had been advocated, and research had been going on for a few years.
Yeah, absolutely.
But it was not mainstream, and it was thought to be, a bit,
and certainly not commercial, right?
Yeah.
Exactly.
And now it's an incredible innovation driver.
Yeah.
At the beginning of this, I would not have said that this was going to be the answer. And I was, like so many, was totally wrong.
Well, it's good sometimes to be wrong. I think, especially in this particular case.
Happens all the time.
Absolutely.
Absolutely. Well, I mean, it is interesting though, and I can see that some, research is pushing forward and coming back in a bigger way. But Mario, when you were talking earlier about your excitement about molecular imaging technologies coming out and how that spurred you on to found BioLaurus, what's exciting you right now? Is there any stuff that's coming out right now that you're like, oh my God, this is amazing?
Well, some of it is looking at the new developments in AI, in artificial intelligence. And it's not simply processing big data.
Right.
When we're looking at data for imaging, these are huge data sets. And the processing of that is, I think, fairly well defined at this point, being able to process and being able to categorize data that then we can evaluate and come up with new discoveries. AI is going to be a facilitator.
Yeah.
It isn't going to do it for us.
No, I think absolutely that's a good distinction to call out, but can facilitate, can scale, can perhaps do that thing that so long ago you were trying to do, which is to speed up the process, yes. Which is so critical.
Absolutely, absolutely. And there's been some areas where that is making a critical impact, and all the way from direct patient care and doing all the background sort of sorting. I know this is going on with brain disease research, where they can gather now data from across the world on patients with given diseases, and be able to begin to use AI to organize what would be trillions upon trillions of bits of information, and be able to begin to work it into a stream and prioritize that for researchers to effectively now get a hold of. So now that they can look at what would be historical data with even tens of thousands potentially of clinical trial participants is I think going to be enormously powerful, because that precision medicine and the way that therapies are being developed for you as an individual, that's the key to the future. We're not all the same, and we don't fit just a population distribution. Those that respond very well to a drug, the average response, and then there's people that don't respond to a particular drug at all.
Right.
Yeah, so we have to really tailor and shape and take that into consideration as we map out not only the final products and the rollout, but as we design these new products going forward. Absolutely. And thinking about going forward, Mario, I mean, here we are going into 2023. If you were to look through this coming year, what are the key things that are going to be on the docket for BioLaurus and also just in the industry in general?
Well, I think this is for BioLaurus, the CRO model is one that the excitement that I have is moving away from those kind of research services to more directly into how do we incorporate our capabilities to be able to process data, to do this analytics, because this is something that this more specialized, and we can collaborate with far more people to be able to investigate how AI is going to help us do that and make this whole process of drug discovery and transition through the preclinical space much more efficient. So I think that, that is where BioLaurus is going. My personal interest is reshaping what I can do and be able to work more directly, advisory services, and becoming involved in one or more biotech companies and specific kinds of programs, particularly in oncology, that I may bring this experience to. So again, that goes kind of with my theme, and that is to always get excited about new things.
Yeah.
The funny thing though is that not everyone can see new things and make it into something that's useful or into an enterprise that can be commercially viable, or to connect it into other things that already exist to make it something that can scale. And so Mario, I mean, certainly it's a great one. I'm imagining maybe in the near future we see a Mario AI. I don't know. Maybe. We'll see how that goes.
We'll see.
Well, it's been a blessing to talk to you, Mario. We've been speaking with Mario Bourdon. He is the Chief Scientific Officer at BioLaurus. Before we go, I mean, if someone wanted to learn more about what you and the BioLaurus team are working on, where's the best place to find you?
Well, I guess our website or my LinkedIn. I'm there on LinkedIn, so that's one good way. And the other is of course to the BioLaurus website, so biolaurus.com.
Mario, thank you so much for joining us today. Mario is the Chief Scientific Officer of BioLaurus, and they're innovating the future of in vivo preclinical molecular imaging by combining study design consulting, study management, and image and analytics. And we've been talking about where he sees the industry going in the future. It seems like it's going to be an artificially intelligent future, which that supports the process of making analytics be processed and utilized more effectively, efficiently, and really bringing new drugs to market in a more positive way. Mario, thank you so much for being on Uncaged today, and we look forward to having you back.
Thank you very much, Ben.
Cheers.
Cheers.